Okay, I assume everyone can hear me. Firstly, thanks to Betty and the organisers for letting me speak today. My name is Jason Clark. I'm Chief Technical Officer at Novalytics. I'm going to be talking about our progress in using the phages produced to GMP, hopefully in future for clinical trials. So just a little bit about the company. We were originally a spin-out from Warwick University. We've been going since 2002, although I only joined a year ago. We're currently based in Daresbury, which is midway between Manchester and Liverpool in the UK, and we're on a science park there. And we are developing phage therapies to treat antibiotic-resistant infections, as many people here are. And our first target is Staph aureus, and we're looking actually, our first indication is nasal decolonization of Staph aureus. And we're doing that using a gel to deliver a phage cocktail to the nose. Now I'm going to try and cover an awful lot of stuff today because Novalytics haven't actually presented for a number of years. So what I'm going to do is I'm going to use a kind of a simplified timeline to kind of put everything in context that we've done and just go through it and use a couple of examples from our experience, although the timeline is generalized. So this is actually more things as they should have been than actually as they were, because if I told you what actually happened, we'd be here till about 9 o'clock again. So, and actually there's not going to be a lot of science in this. There'll be probably more answers than questions, more questions than answers by the end of it. But I encourage you to come up and speak to either myself or John Harcastle, our CEO. He's here as well. We're very happy to chat. This is really just to let you see what we've done and say, please come and talk to us. So the first step was obviously isolating phages and defining our phage cocktail. Because I'm going to focus more on the process development, all I want to say about this is, right from the very start, we thought, we want to produce this cocktail to GMP, so let's try and minimize the number of phages that are present. So we worked quite hard on this, because the more phages you have, the more expensive it is to produce the cocktail. So we've come up with what we think is a fairly simple cocktail that kills 93% of the clinical isolates worldwide that we've looked at. All the phages are fully sequenced. There's no toxin genes, obviously, they don't transduce, and we can produce them commercially. So we know what range of pH they're stable in, we know what salt concentration they're stable in, and we know they're not susceptible to the kind of shear forces you see in commercial development. So we think we can scale them up industrially. The next two steps are kind of related. Obviously, we could purify them before we could do the efficacy tests, but the kind of purification I'm talking about here is the kind of purification you can scale up. So not gradient ultracentrifugation, but the more standard techniques you would use for purifying viruses. Chromatography, ion exchange chromatography, size exclusion chromatography. You might also want to look at filtration, or even PEG precipitation can be used in GMP. And I can't say much about our actual purification method, but it is column-based, and it does give us phage of a purity sufficient for topical application, and it does seem to be very scalable. I mentioned we deliver our phage in a gel matrix, and this was actually a matrix that we developed ourselves from scratch. So initially we looked at 60 different gel formulations purely on the basis of the physical characteristics. How stable were they at different temperatures? How did they respond when you rubbed them on the skin? We picked the best 20 and tested how active our phage were in the gels. So were the phage stable, and could the phage diffuse out of the gel and actually come in contact with the bacteria in kind of as it would in the natural environment when we're using them for treatment. The three best gels were tested in a sheep mucosal model. So this was an in vitro assay where we use cell culture, we colonized the mucosal cells with staph, and then we treated them with either the three different phage preparations or mupirocin, which is, or was actually Bactroban, which is a commercial product that you can buy. It's based on mupirocin. And of the three gel products, Nov 012, as we call it, was considered significantly more active than the other two gel products, but in this model system it was actually more active than the Bactroban treatment. And as I said, I'm not presenting all the results we have, but based on this and other data that we have, we decided to progress with this gel formulation. And obviously all these have the same phage cocktail in. So we've got our phage cocktail, we've got our delivery matrix, we think we can purify and manufacture everything in-house, but then we need to speak to manufacturers. And this is actually not a trivial issue because this is someone you're going to be working with for a number of years. So you have to have confidence that they can do what they say they do. And if you can find someone who's worked with phage before in the past, all the better. So then you have your standard technology transfer steps, health and safety analysis to ensure that they're happy working with your phages. And even though everyone here probably knows that phages are safe, you need to prove that to your contract manufacturers. You'll transfer your assays, you'll transfer your process that you've got thus far, and you'll adapt it to fit into the equipment that's present in your manufacturing site. That might involve more assay development and more process development. At this point, you will actually start to put everything together and do an engineering run of the phage. So this is where you try and mimic as closely as possible what will happen at the GLP stage, at the GMP stage, sorry, but it will be done to GLP, so good laboratory practice. And this is just to ensure that you can scale everything up and everything works as you expect, but it doesn't incur the cost of GMP production. And there are two further advantages of doing an engineering run at this stage, is you can actually start your stability testing early, because when I get to down here, you'll see that there's stability testing after the GMP manufacture as well. But if you can convince the regulators that your engineering run is comparable to your GMP run, you can start stability testing early and you can significantly reduce the timelines later on. Another thing you can do with an engineering run is preclinical toxicology. And this is actually what we did. We did an engineering run, made a batch of gel and used it for preclinical tox. And just again, to go through this quite quickly. There are 5 different studies actually involved in the preclinical tox package, a variety of different model systems, different doses, different dose schedules, and I don't think it will be a surprise to anyone here that there were no signs of systemic toxicity, and there was no irritation in any of the models either. The one thing I would add about this table is, and this is just to give you an idea of the kind of timelines that I'm talking about here, because I'm rushing through everything quite quickly. We first approached the MHRA, who are the regulators in the UK, with 4 of these studies. And they said, that's all really good, but we'd quite like to do a repeat dose study as well. So we then had to take a step back, go away, do a repeat dose study. And of course, the repeat dose study cost twice as much and took twice as long as all the other studies combined. As is the way with these things. So in total, the preclinical tox probably took us about 9 months. We were doing other things at the same time, obviously. Okay, so we're quite happy now that we can produce the things on a larger scale. We've done our preclinical tox, so we're confident as we can be that when we do move to GMP manufacture, nothing's going to go horribly wrong. And that's quite important because when you book a space in a GMP suite, you'll typically have to leave about 3 months, or allow for 3 months for them to have a space free in a GMP suite. So you don't want to book it too early because then something might happen and you've paid a deposit and you've lost a lot of money. But at the same point, you don't want to do it too late because you're working on quite tight time schedules, so you need to try and compress everything wherever possible. And just a little aside for a moment, because speaking to people both at this conference and outside of the conference, it's not always certain that everyone knows what GMP manufacture involves. So I did have a couple of slides which were far more complicated, but last night I went back to my room and simplified it down to just a couple of key points. Basically, GMP is validation and consistency. What you want to be able to do is say, within the parameters we have defined, the batch that we have made today will be the same as the batch we make tomorrow and the batch we make in a year. GMP is not a measure of purity. You always have to go in with set limits of contamination, for example, in terms of host cell DNA, host cell proteins, endotoxin, or protein A, for example, depending on what bacteria you're working with. In theory, you could produce a phage preparation to GMP that had 10% sodium azide. And you could produce it to GMP so you know that the next time you make it, it will have 10% sodium azide as well. You would not be able, I assume, to get ethical approval for a clinical trial using that though. What it will help you do is prove a lack of adventitious agents. If you have a good GMP procedure, you should be able to convince the regulators at the end of it that you have no TSEs, contaminating viruses, and the like. And just to say, when we spoke to the MHRA about this, they indicated that in the past they have given special dispensations for phage therapy trials in the UK. When we spoke to them, they were fairly blunt, saying that, at least in our case, this wasn't going to happen, and we did need to produce the phages to GMP if we wanted to do a clinical trial, and there was no way around that. So we're happy with everything. Now we actually get to produce our clinical batches. And this brings all the experience together, everything you've learned over the past couple of years in our case. You make your phage, and then you mix the phage with the gel and add it to the tubes. And just to give you an example of the kind of level of detail we're talking about at GMP, this is the index page with a few deletions, an index page of a batch manufacturing record for the fill finish step. So this is just mixing the phage with the gel and putting it in tubes. And you can see that it's 108 pages long, so it doesn't include any of the phage purification, it doesn't include any SOPs or the standard quality management documents that you would have. And it's basically 108 pages of this kind of thing. So everything's rigorously detailed, signed, countersigned. And you can imagine that developing this and doing it for every batch is time consuming and pretty expensive, and you can start to see where all the money for GMP manufacture goes. Okay, we're almost there. Once you've actually made your phage gel, in our case, the two remaining things that you need to do are bioburden testing. So at some point you will have defined the level of bioburden in your product, either sterile or some other grade suggested by the European Pharmacopoeia. So you need to test and prove that you have less than those levels of contaminants. And, as I mentioned, you do need to do stability testing on your final product. Hopefully you've learned enough from this that you can get away with a month of stability testing here, and during that time also you would look to get it signed off by a qualified person. So that's just the final stamp to say someone has reviewed all the documentation, they've taken a couple of weeks to do it, and they think, okay, this is good, they can use this in a clinical trial. And in a vastly underwhelming fashion, this is what a tube of GMP-produced phages looks like. The only difference is we did remove the label before taking this picture because you wouldn't believe how long it took us to actually define what was on the label. So we thought, for the classic commercial reasons, we would remove it before we put a picture up. So you have to take my word for it, but that does actually contain phages produced to GMP. And if you speak to us afterwards, we'll hopefully be able to prove it. And just in the last minute or so, so that's a whistle-stop tour of what we've done production-wise. There's so much I've missed out. It's unbelievable. But just to highlight, we have also designed our clinical trial for nasal decolonization. We've completed the investigator's brochure, submitted it to the ethical committee, and we do actually have ethical committee approval to begin this trial. At the same time, we've submitted our IMPD, Investigational Medicinal Product Dossier, which is the fairly substantial document you need to submit to the UK regulators whenever you want to start development of a new therapeutic product. To cut a long story short, there was a lot of, well, blood, sweat and tears, for want of a better expression, and quite a few meetings with the MHRA, but the last time we met them earlier in the year, they indicated that they were happy with all the changes that we had made, and they had no further comments on the IMPD. Commencement of our clinical trial is imminent. I can't go into any more details than that, but we might if you speak to us afterwards. And just to highlight, this is really several years worth of effort. We've not presented for a long time, so I've tried to summarise everything into 15 minutes, and I hope you could follow it. I was going to talk a little bit about some of the regulatory considerations, but as I expect, I'm out of time, and I think they've been covered quite nicely by the speakers earlier in the earlier session and the session today. So I'll just leave you with a little cartoon to highlight one point. Whenever I stole this from the XKCD website, by the way. Whenever you read a report that phages kill bacteria in a petri dish, remember, so does a handgun. We all want to be able to use phages in humans, whether it's using GMP phage preparations or bespoke phage cocktails. And we have to, the more people working on it, the better. That's what we believe. So please come and talk to us afterwards. Thank you.
Thank you, Jason. You have questions?
Philip?
2 questions.
Okay.
Can you tell us what the gel is? Number 2, have you tried to grow the phages in the presence of the gel?
We cannot tell you what the gel is. We might be able to soon, but that's basically know-how just now, so we can't tell you what that is. We have not tried to grow the phages in the presence of the gel. We know the phages are stable, and we know that they can diffuse out of the gel, but we've not actually tried to grow them in the presence of the gel. And that actually, like I say, if you come and speak to me afterwards, there's quite a lot I didn't discuss about the gel, and I can go into more detail if you come and speak to me.
Did you go only for a national registration?
The question was, did we go only for national registration? And the answer is yes, at this stage.
The EMA recently asked for all new anti-infectious products to go for a central procedure.
We've been going through this for quite some time, and
the key thing really is you need to start local.
yeah, I mean and I think this is your experience as well. You need to start local because you're speaking to the local regulators first before discussing it with the EMA.
Thanks.
And it's, we're quite a small company. We're quite stretched as it is.
If you get MHRA, it makes the process, theoretically the process in Europe that much more straightforward because you've got leverage. But you really
Just repeat the question.
Repeat the question.
So it was a continuation of the question, did we just go for national approval, basically? And the answer was yes. We have only spoken to the MHRA so far.
Yeah. I mean, bear in mind, we went to see the MHRA 3 times at critical points in terms of scientific advice, and they were feeling their way as well. What you don't want to do is make it even more difficult by then saying, well, how's it going to be accepted in Europe and obviously given where we are in the States. So if you start with one reasonably well-respected regulatory agency and then shift outwards was our view, the best way of getting it through.
I think that's right. I think the regulators are also wondering how to best handle phages.
The other thing about it is that the regulatory authorities are not difficult. They're just used to looking at how to stop drugs coming through that shouldn't do. So that the start point is, we know it's not a drug, but we're going to treat it as if it were, and we will apply the same kind of criteria.
The example I like to give of that is that we had to convince them quite vigorously that we didn't need to do a dose response for the toxicology, because with phage, a dose response is a bit meaningless. But we had to convince them of
PK/PD for that matter.
Yeah. Oh, Sandra.
Did you discuss with them about having a fixed...
Nobody can hear you.
Where is the microphone, sir? I was just curious, when you spoke to the agency, did you have like a fixed mix, or are you going to be able to exchange phages over time, or what was the feedback on that?
Yes, it's a pertinent and very pertinent question. We have raised the issue, and the thing when we spoke to them they were most worried about was the total number of phages rather than the exact makeup of the phage cocktail. But we have not specifically approached them about modifying, replacing one phage with another phage. That's the position we would like to get to at some point. So I mentioned the flu vaccine, that's the most sensible way to use phages in this particular fashion. But I can't give you a good answer on that. They seemed receptive is all I would say. We've not asked them in detail.
The big thing again is active substance. Is your active substance the individual phages in the cocktail or all of them?
So Jason, do you have to provide them with a chemical analysis of basically what's in there at a chemical level?
I would not maybe say the chemical level, but what they are concerned about is not the phages themselves almost, but they are worried about host cell protein, host cell DNA, and it's as much from the point of consistency as them being worried about anything. They like you to prove consistency more than anything else. They want to know that the batch you've used for your preclinical talks is the same as the batch you're using in your phase 1 trial. So not necessarily a chemical level.
If you define it in terms of chemical composition or protein composition, you can change within certain parameters.
Yeah, sorry.
People can't hear.
Oh, sorry. Mark was just asking if they asked us about the chemical composition of the phages that we made. And the answer is, I guess, yes, up to a certain point. They wanted to know host cell protein, host cell DNA, and obviously you would do an SDS page gel. So fairly crude determinations of the bands look the same as they did before. Because of the purification method we're using and because of the materials we're using, there's nothing in there that should be dangerous. There's no mammalian cells to worry about viruses creeping over. So they're actually relatively relaxed. If you can show consistency, they're not actually too worried about anything dangerous.
Thank you, Jason.
Thank you.